Thank you for giving me the opportunity to speak with you all today. Typical clinical genetic evaluation is performed in a stepwise fashion where one test is sent per visit and multiple visits may be required for it to be comprehensive. This can take years to complete, places an unnecessary burden on individuals and their families. It's expensive and surprisingly is diagnostic in fewer than 50% of individuals. In the Miller Lab, we're interested in using long-read sequencing to improve clinical genetic testing by shortening the diagnostic odyssey and reduce barriers to accessing comprehensive genetic testing. Long reads simplify the problems encountered from using short reads for exome or whole genome sequencing, such as the assembly of repetitive regions like this one that I'm showing here, by simply being longer than the length of many of these regions. Thus, longer sequencing reads improve our ability to both capture and resolve structural variants. And additionally, we can simultaneously detect epigenetic modifications in the reads. There are many challenges with our goal to improve clinical testing, but a specific one is how do we prioritize and filter the structural variants we can now detect using long-read sequencing that couldn't be detected in the short-read sequencing. And so to address this, we turn to the 1000 Genome Project sample collection. This has been an incredible resource for understanding patterns of normal human genetic variation from over 3,000 individuals. And we can expand the scope of this resource by using long-read sequencing to further investigate previously difficult to sequence regions, complex structural variants, and simultaneously evaluate epigenetic modifications. The work I'll describe is from the 1000 Genome Project ONT sequencing consortium that is led by our lab as well as Evan Eichler's lab at the University of Washington. We've sequenced close to 200 of our 800 samples with Nanopore sequencing. All of the data that we are generating is publicly available on AWS, and I'll be showing results from our analysis of the first 100 samples, which are represented in the darkest shading in this plot that I'm showing here. So these first 100 samples were sequenced with R9 flow cells to at least 30x coverage and have read N50s of at least 40 kb. So to get the very high molecular weight DNA for achieving these high N50s, we grow lymphoblastoid cell lines obtained from Coriell, perform the extractions ourselves, and then sequence each on our ONT Prometheon. And after sequencing and base calling, we perform multiple types of analysis. One thing we use is the NAPU, formerly known as the card pipeline, for generating haplotype-resolved assemblies and phased alignments. And today I'm going to focus on our methylation analysis and our evaluation of repeat expansions. And I'll be starting with what we've done looking at the repeat expansions. So as I've said, because long reads can span complex regions like repeats, such as the one shown here, we're able to recover not just repeat length, but also the sequence-specific motif composition. So in this example, if we just looked at our reads in IGV, we would see there is an inserted sequence in each of our reads. And we are able to generate an assembly for each haplotype from our reads, resolving the two allele sizes at this locus. And from each assembly sequence, we can use a genotyping tool such as VAMOS from Mark Scheisland's lab at USC to count the number of total motifs and determine the actual sequence of each unit for each allele. Knowing both the repeat length and motif composition can have significant clinical relevance due to sequence-specific nature of some repeat expansion diseases, such as the one I am simulating here at RFC1, which we heard about yesterday, which is associated with the disease canvas. And when we look at this complex locus in the 1000 Genome Project samples, we found that 5 of the 100 individuals analyzed had an expansion with 3 of those alleles over the threshold of what is typically considered pathogenic. And most importantly, the smaller bar plot are the results for the same samples using expansion hunter estimates based on the short read data. And what we find is that a larger allele than the normal allele size can be regularly detected, signifying that there might be an expansion allele, but it still significantly underestimates the size of the expansion. You can also see that we can evaluate the motif content, identify the known pathogenic motif, and see interruptions in the motif. Importantly, whether these are real pathogenic alleles in this reportedly healthy population or an artifact of the cell culture process is unclear. Regardless, this demonstrates our ability to identify repeat expansions using long-read sequencing and helps us think about how we could analyze these data in the clinical setting. We want these data to be accessible to everyone, so as I demonstrated yesterday in my pitch, we're making tools to make interacting with this data easier. This is that browser tool that I developed that lets you navigate genotype data at over 60 loci, and this will be a useful resource for quickly comparing results of an individual to that found in a healthy population. So I've shown some of our preliminary results using haplotype-resolved assemblies to analyze repeat expansions, and now I'm going to switch gears to talk about methylation. So long reads are able to be phased into parent of origin haplotypes, which is what I'm showing here at KCNQ1, which contains an imprinted locus highlighted by the CPG island, which is associated with Beckwith-Wiedemann or Russell-Silver syndrome or Silver-Russell syndrome. And when we look at the methylation information contained in the reads, we see the expected pattern of methylation. One haplotype is methylated or silenced, and the other is unmethylated or active, which is the one in blue. And we can quantify the methylation patterns at loci associated with disease. And we find that across 100 samples, broad patterns of differential methylation that we are expecting are preserved, which means we should be able to develop tools for identifying methylation outliers in this data. To help do this, Miranda Zaleski from our lab developed Meow, or a methylation operation wizard, which is able to identify differentially methylated regions within individual samples. And when we iterate this through the first 100 samples, we identified samples with DMRs unique among the cohort, including this one at SLC2983. You can see that one haplotype of the sample is methylated, while all of the other haplotypes are unmethylated. And we can also use methylation data to characterize larger patterns of methylation, such as X chromosome inactivation. In this sample set, we find both patterns of random X inactivation, where the fraction of reads methylated per haplotype is equal, as shown here, as well as evidence of skewed X inactivation, where one haplotype is preferentially methylated across the entire X chromosome. In fact, we found that among our samples with 2 X chromosomes in the first 100 that we sequenced, 39% of those had skewed X inactivation. This is likely due to passaging in the cell culture process, and this finding demonstrates both the need to be aware of skewing in these samples and the limitations that could be associated with using immortalized cell lines. So in summary, I've shown how we use long-read sequencing to improve our understanding of repetitive regions and identify novel patterns of methylation at specific loci, as well as across whole chromosomes. But what we're really excited about is making novel discoveries in previously inaccessible regions in these samples, and as well as using these to identify variants in patients who have remained unsolved for so long. And with that, I'd like to thank my funding sources, everyone who has contributed to the 1,000 Genomes ONT Consortium, especially the Eichler Lab. This report we've received from ONT, members of both of my labs, and I'm happy to take any questions.